Okay, let's go then. Okay, I will just start with a quite simple observation. Here you can see the largest family, I think the largest in the world, is the British family, and they got 21 children. It's the same couple, they got 21 children. And what is very striking here is that every one of these brothers and sisters are each different from each other. How is that possible? And what we observe, of course, in humans, is also true in other animals or plant species. And of course, you are all aware of that. This is because of meiosis, or thanks to meiosis. So whatever eukaryote you can think of, they go through sexual reproduction thanks to meiosis. Meiosis shuffle chromosomes at each generation, making new offspring unique. You are unique thanks to meiosis. You are different from your brother and your sister because of meiosis. Because at each generation, meiosis pick up one of the two chromosomes in each pair, plus recombine those chromosomes to make new unique chromosomes. So for example, your chromosomes have been assembled very likely like 9 months and a few weeks before your birth on the male side, and between 20 and 40 years before your birth for the female side, when your mother was an embryo. And okay, this is a relatively simple mechanism. I guess all of you, or many of you, have bad memories of learning the stages of meiosis, prophase, metaphase, even leptotene, zygotene, all of this. But I'll try to have you with me through the mechanism of meiosis, and I hope that won't be a bad memory. So there's two divisions following a single replication to achieve the reduction of ploidy, and the key event of shuffling the chromosome occurred during the first division. My talk will be split into two parts: how we can play with meiosis, tinker with meiosis to increase recombination, and oppositely, how can we tinker with meiosis to abolish recombination. So let's focus now on meiotic crossovers. I've told you already that crossovers make unique chromosomes. Thanks to crossover, some combination of alleles are possible. So for example, here in this very historical example, this combination of phenotypes is possible only if a crossover occurred right in between those two genes. There's a second function of crossovers which is typically less well known, which is a physical, mechanical function of crossovers. Crossovers connect chromosomes during meiosis, and this is required for proper segregation of those chromosomes at meiosis. So with the lack of crossover, you have a missegregation, and for example, lack of crossover is a primary cause of missegregation in human, of miscarriage, and of trisomy 21, for example. So this is because of a lack of crossover most of the time. And when we look at the number of crossover per generation, there are two striking observations. The first one is that you observe at least one chromosome on each pair of chromosome, one crossover on each pair of chromosome at each generation. This is what we call in the field the obligate crossover. This is obviously linked to the mechanical function of crossover to connect chromosomes together. But when we look eukaryote-wide as a number of crossovers, we don't see many per generation. Typically 2 to 3 per chromosome per generation. And to convince you of this, I have made up that plot. So let's take a minute to go through it. So this is a compilation of data from many different eukaryotes. Each dot is a chromosome, and each color code is a species. On the x-axis here, you have the physical size of those chromosomes in megabase, and that's a log scale. And here, the estimated number of crossover derived from genetic maps. The first thing I'd like you to see that first, there's nothing here. So there's no species with less than one crossover per meiosis. But second, and I think that's quite striking, most chromosome eukaryote-wide receive between 1 and 3 crossover per generation. And this is completely independent of the size of those chromosomes between different species. Within one species, you have a correlation between the size of the chromosome and the number of crossover, but not between species. So for example, here on the left, you have button mushroom or trypanosoma. The size of the chromosome is roughly 1 megabase, and they receive 2 crossovers. And on the right, you have barley and opossum, and the chromosome are 1,000 megabase, so 1,000 bigger, and still receive roughly two crossover per generation. So this is really a striking distribution of crossover towards low number. Of course, always in biology, there are some exceptions. So two notable exceptions here are Cerevisiae and Pombe, two single-cell yeast with a much higher number of crossovers than any other average species. We don't have that many species for which we have both a chromosomal assembly and a good genetic map. So we can only speculate of why we have those exceptions. So for example, here it's maybe because those single-cell fungi have a different optimal of recognition levels than other species. Another striking exception is honeybee here with much higher crossover than most eukaryotes. Here we can also speculate it could be due to the specific mode of reproduction of honeybee with a single female and haploid males that can also end up in a different optimum of recognition levels. Chicken and birds in general also tend to have some chromosome with high number of crossover, and in that case, it's probably linked to the very weird distribution of the chromosome size in that species. Within the same species, you have chromosomes which are roughly 2 megabase and chromosomes which are roughly 200 megabase. So you have a 100-fold difference in size within the same species. And likely to ensure crossover on small chromosomes, then you end up with more crossover on the large one. But most of that is just speculation. And finally, cat is quite high. This is the highest I've seen so far in mammals. Why cats have more crossover than others? Honestly, I have no good idea whether that just cats are crazy. Okay, back to my favorite model, Arabidopsis, and actually all plants are here in the middle of typical species with roughly 2 crossover per generation. Let's go now a bit through the minimum background to understand the following work, which is what we know about crossover formation, how that works. I've just put that picture because to summarize how it works, it's just breaking and repairing DNA. This is the process. So you start recombination by breaking your DNA in multiple pieces. So here you have the representation of the two homologous chromatids, both double-stranded, and you have double-strand break here. And those double-strand breaks are repaired using the homologous chromosome as a template. And this leads to crossover formation. I will just ask you to remember the existence of these two proteins, DMC1 and RCT1. This is really two magic proteins that invade, that catalyze the invasion of the homologous sequence by the broken chromosome. We can look at those proteins with cytology. That's how we estimate the number of breaks in each cell in Arabidopsis here, roughly 200 in Arabidopsis. It varies from few hundreds to a few thousand depending on the species, but few hundred is quite typical. And from those few hundreds double-strand breaks, only a few of them become crossover. We and others have described in the last decade 2 pathways to form crossovers. The main one called class 1 crossover, depending on those proteins, and the second one, which is very minor in wild type, making roughly 9 and 10 and 1 crossover per generation, roughly 10 altogether in Arabidopsis. And again, we can stain some of those proteins along chromosomes. So what is, okay, we may wonder, what? 200 breaks, 10 crossovers, where are the others? All the others are repaired without making crossover. So there's a very large number of precursors for eventually a low number of crossover. So we end up with that observation, or those observations. Crossover number is very low in most species, while the number of precursors is much higher. So why having so few crossover in most species? How is that possible? So I'm a geneticist, so basically I have a single answer to any question, which is let's run a genetic screen. So we set up a screen specifically to identify the genes and the mechanisms preventing crossover formation. So this is a classical trick. We use a suppressor screen. We started with mutants in which crossover formation is defective. And so you can see here, because of that, some chromosomes are not connected to each other because you don't have this physical link between chromosomes. Then you have misirrigation of these chromosomes. Most gametes are aneuploid, and then the plant has a very strongly reduced fertility, which is pretty useful for us because this is visible just by looking at the plant in the greenhouse. They have shorter fruits, which is easy to score. So we apply the secondary mutagenesis on those mutants, looking for plant with restored fertility, which is easy to score, as a proxy for restored crossovers, with the idea that if we have this, this means that we have disrupted an anti-crossover gene. So we've done that at a quite large scale. This was in the greenhouse in Versailles, in France. Most of those plants have very strongly reduced fertility, but a few ones, the suppressors, have really increased fertility, and as you can see here. And this was a very specific screen. Every single mutant we identified with increased fruit size had increased crossover number. We've done that at a quite large scale now, screening thousands of lines, which mean hundreds of thousands of plants, identified 8 genes so far, defining 3 parallel pathways that prevent crossover formation. So this is a huge amount of work among the years. I will come back a bit later on what are those pathways, and focusing on the last one, which is the less expected, I would say. Okay, so we have these mutants with increased fertility, increased crossover number according to cytologies, and we use a bunch of tools to confirm that. I'll show you one piece of data here, because this is a very powerful tool and very beautiful tool developed by Gregory Kuppenhaver here in the US. This is based on segregation of transgenes that confer fluorescence to pollen grains, and those transgenes are linked here. So just by looking at the distribution of colors, you can score a combination. This is pretty powerful. So we've done that with several intervals. I'll show you one. This is recognition level in the wild type in this interval, and this is the same measurement in the mutants of the different pathway. So we have a very clear, large increase of recombination in those mutants, ranging from 2 times to 4 times, or 6 times in that example here. Actually, this one is very specific. This is in a single interval here. This combination of color is possible only if you add 4 crossovers in the same cell in this tiny interval here. So this is really completely different from a wild type. Never see that in a wild type. Okay, next we ask ourselves, okay, where are these new crossovers coming from? So the first simple hypothesis, maybe they do even more breaks and then making more crossovers. So we check that by using, again, this marker, DMC1 foci, and estimated the number of breaks. No obvious difference. So it's clear that in those mutants, we have more crossovers from the same number of breaks. Then we ask ourselves, either the class 1, class 2 pathway, both, or something else. I show you one piece of evidence, which is the staining of the class 1 crossover, which is specific to class 1 crossover. You have roughly 2 per chromosome of that one, because that's the main pathway. We don't see any difference between the mutant and the wild type. So the class 1 pathway is not modified. This and many other evidences, I won't spend too much time to present you today, led us to the conclusion that in all those mutants, we have the same phenomenon, which is a very large increase of the normally minor class 2 crossover. So in wild type, it's very minor pathway. It's mostly class 1 crossover. In those mutants, the class 2 crossover is increased so largely that now it outnumbers very largely the number of class 1 crossover. And this is from the same number of breaks. So we changed the balance between non-crossover repair events towards class 2 crossovers. Okay, let's go a bit for a couple of slides into the molecular mechanism behind those mutations. So just one slide on the 2 first pathway. This is published work. Both of those pathways rely on helicases. Well, one is FANChem, the other one is Bloom or RECU4. Those helicases are very conserved in eukaryotes, from humans to plants. It has been shown with the human version or the mouse version that those proteins can disrupt this kind of intermediates, which is an intermediate towards crossover. So it's quite easy to put forward an explanation how those proteins can prevent crossover formation in wild type. Let's focus now on the third pathway, which was less expected. So we had two genes in that pathway called FIGL1 and FLIP. FIGL1 is a AAA ATPase. This family of proteins form typically hexamers and are able to target other proteins to disrupt their 3D structure using ATP. So they are typically negative regulators of other proteins. This one, FIGL1, is quite conserved in eukaryotes. You can see here the red dots throughout the tree of life of eukaryotes. It has been lost in fungi specifically, which is interesting, but it's present in all plants and all animals. It has been shown to interact with RAT51 and DMC1, or RAT51, one of the two proteins that invade the homologous chromosome in human, giving a first hint about the function of that protein. The second one is the pathway, FLIP1, no clue what that protein. The second tells nothing. We have no data of interaction. We're just in the dark with that protein. It is conserved also well in eukaryotes. The structure, the function of that second protein, no idea. So we looked for partners of those two proteins. So we started with FIGL1 with the protein purification in vivo, in cell culture. So when we used FIGL1 as a bait, we got a single hint, which was FLIP1. Okay, good. We started with FLIP1, and we got a single hit, which was FIGL1. Okay, good conclusion. Those two guys make a multimer or a dimer or higher level in vivo, but what do? No more information. We showed with this to hybrid that this is a direct interaction using Arabidopsis protein, and showed that this is also true for the human version of those proteins. So very likely this heterodimer is conserved throughout eukaryotes. So as I said to you, that we have suspicion that it could act here at this very important step of recombination, which is the invasion of the homologous chromosome catalyzed by DMC1 and RAT51. We showed, again, using these two hybrids, that indeed FIGL1 and FLIP can interact with those two proteins, reinforcing that hypothesis, both in plants and humans. And then we looked in the mutant at the behavior of DMC1 and RAT51. And the number is not really modified. We have roughly the same number of foci here and here, but the dynamic is different. We can stage somatic cells, and actually in the mutant, DMC1 and RAT51 appears earlier and stay longer. So there is a change in the dynamic of that protein. I will introduce one last gene, which is BRCA2, very well-known gene in cancer biology, because this protein or mutation in the corresponding gene in humans is one of the first or the most prominent predisposition to breast cancer in women through the role in DNA repair. And this protein is required for the loading of DMC1 and RAT51. That's how it promotes DNA repair. And that's the same in Arabidopsis. If we have a BRCA2 mutant, those two proteins, RAT51 and DMC1, are not loaded on chromosomes. And consequently, we have also markers for interaction with the homologous chromosome. Consequently, because of failure of loading of those two proteins, there is no interaction with the homologous chromosome. When we combine BRCA2 with FIGL1, then everything is back. We have DMC1, RAT51, and interaction with the homologous chromosome. So we end up with that model that we propose as a new regulating step of recombination, conserved from plants to mammals, which is we have a balance between pro-repair factors, BRCA2, and anti-crossover factor, FIGL1 and FLIP1, acting on this very step catalyzed by RAT51 and DMC1. So just to show how, using Arabidopsis, we can learn new information about how recombination is regulated in eukaryotes. Okay, let's switch to a second part of my talk, which is asking a different question. We have those mutants. How far can we push recombination playing with those mutations? So what we have done is to combine those mutations together in a hybrid with two strains of Arabidopsis so with 1 or 2 or 3 of those mutations, and looked at segregation of markers in the progeny to estimate recombination levels. This is the total genetic map or total number of crossover in wild type, a bit less than 10 altogether, and this is the result in different mutants. I will focus only on 2 of these results here. The single RECQ4 mutant has a 4-fold increase in recombination genome-wide, and the champion is a double mutant FIGL1 RECQ4 with almost 8-fold increase in recombination. This is a huge increase. We combine the 3, maybe we can go higher. Actually, we don't go higher, so it suggests that playing with those 3 pathways, we have reached a kind of upper limit. Maybe we have used up all the double-strand breaks which are available, or something else, we can speculate. But for sure, we cannot go higher with those 3 mutations. But still, 8 times, this is crazy. And I have a few slides to convince you that this is really unbelievable. It took me some time to digest that information. Here we have wild type genetic map. Many of you, I guess, are very familiar with this kind of maps. And this is a map in the mutant at the same scale. This is a huge increase of the size of the map. If I go back now to my plot with the chromosomes of many different eukaryotes, the wild type Arabidopsis is here, and the mutant is there. So this is almost the highest recombining eukaryote on Earth now, just combining 2 mutations. Just 2-point mutations. One last piece of information. Here, this is a sequencing of the 4 product of a meiosis, the 4 pollen grains. So you can see crossover, which are reciprocal events. Here, 2 or 3 on chromosome 1, 2 on chromosome 2 on this specific meiosis in wild type. 10 in that case, very typical. And this is what we observe in the mutant. In that example, we have 62 crossovers, 19 on single chromosome 5. And you can see how chromosomes are just crumbled in those mutants. So obviously, you have new combinations of allele here, which is not possible to get in a single meiosis in wild type. Okay. Does that affect fertility? We have this incredible increase of recombination. Maybe someone will say, yeah, meiosis will be screwed up and we'll have completely sterile plants. No. Just this simple assay containing seeds, we don't detect any significant difference in fertility. They produce seeds normally. Using a bit more sensitive assay, we detect some defects. Like here we have, if you look at the viability of pollen grains, in some of those mutants, you indeed detect some death of the pollen grains. And if we look at meiosis, we can correlate this low level of pollen death to a low level of meiotic defects, which are chromosome bridges and chromosome fragments. So these are signs of failed double-strand break repair. So I would argue that this tiny decrease in fertility that we observe in those mutants is due to this failed repair, double-strand breaks, rather than due to the very large increase in crossover number. And crossover number by itself doesn't look to have an effect on fertility. Next, okay, we have this very large number of crossovers. Where are they along the genome? So we map them. Here you see the 5 chromosomes of Arabidopsis. The black line here is local crossover level along the chromosomes. What is clear and observed in all eukaryotes is an abolition of crossover formation at centromere on all the chromosomes, and you have variation along the arms of the recombination level. This is the same experiment in the champion mutant, RECQ4 FIGL1. You have this huge increase everywhere of recombination, but not at centromere. Not in the center of centromere where recombination is abolished, but not either on both sides where recombination is active in wild type, but is not really increased in the mutant. So we conclude from this result that something else must prevent crossover formation next to centromere, at and next to centromere. And this is the next challenge in the field, to identify the factors that prevent crossover formation next to centromere. Okay, all of this, what I've shown so far, is Arabidopsis. Many of you say, oh, Arabidopsis, we don't care. We want real plants. Okay, let's go for real plants. We pick up 3 species. pea, tomato, and rice, that span more or less the diversity of crops, and just look at the REQ4 mutant, the most effective in Arabidopsis. We start with rice. We used, it was just before CRISPR, and we initiated that experiment, so we used just regular insertion in the REQ4 gene in rice in 2 different strains, allowing us to make F1 hybrids, mutant and wild type, in which we don't see any defect of fertility. And then we, again, made genetic maps in both wild type and mutant, looking at segregation of markers. This is the map in wild type of the 12 chromosomes in rice, and this is a map in this single mutant. Again, we observe a very large increase of recombination levels along all the chromosomes by a single point mutation. Well, single insertion in that case. This is on average a more than 3-fold increase genome-wide. If we look again at the distribution of events, we have the exact same observation than in Arabidopsis, that we have a large increase along the arms, but no increase at centromere. So the observation is very similar. This is not the solution this is a solution to increase recombination, but not the solution to unlock centromeres. Pea and tomato, briefly, same thing. We look at the 7 chromosomes of pea and 2 of tomato, but that's enough to show that we have the same increase everywhere. So it looks to be universal. We have an increase of recombination on all the chromosomes using REQ4 mutation in Arabidopsis, rice, pea, or tomato. So it looks to be a universal anti-crossover factor in plants. So conclusion of this larger part of my talk, 3 pathways actively limit crossover formation. Meiotic crossover can be largely increased without a huge effect on the phenotype of the plant. So this could be a tool for plant breeding to improve power of mapping, to improve cuttile detections, everything based on genetics then can be improved using those mutants. One key conclusion is that those mechanisms, those 3 pathways, don't act or act redundantly with something else at centromere. So we still need to identify those factors at centromeres that prevent crossover formation. And maybe the most intriguing question is, why having so few crossovers in most eukaryotes? It's so easy to increase crossover with no immediate defect. Why having so few? So I will argue, and many others have argued, that very likely there is an optimal for adaptation of the recombination level. I think some recombination is good. You can create new combination of alleles and adapt to new environment, invade new niche, whatever. But having too many crossovers is likely not a good idea, because you shuffle combination or you break combination of alleles that you have selected in the previous generations. So suggesting that there is an equilibrium and an optimal level of recombination, which is actually quite close to the obligate 1 crossover per chromosome, ending up of an average about 2 crossover per chromosome. Okay, let me switch to the second part, which is how can we tinker with meiosis to obtain the opposite, to obtain clonal reproduction, to abolish shuffling, to abolish recombination. This is a mechanism called apomixis that exists in the wild, the production of seeds, but those seeds are clones of the mother plant. So that's derived from sexual reproduction because you have seeds, but shuffling and genetic variation is abolished. This is relatively common. Several hundreds of species have been described as being able to have this mode of reproduction, but it's very largely absent in crops. And its introduction in crops could be really interesting. And why is that? Because you are aware that we use a lot in plant breeding the hybrid effect, that F1 hybrids compete the parents. But the downside of this is that you have to produce F1s every year, because if you harvest simply you seeds on your F1 hybrid because of recombination of meiosis, then you have segregation and you lose the gain of the F1 hybrids. If we will be able to introduce apomixis in those F1 hybrids or any complex genome, complex assemblies you can think of, then we can propagate those elite genome just by, or elite strains just by harvesting the seeds and propagate them infinitely. Some people have described this introduction of apomixis as a holy grail. So a few years ago, we decided to go for the quest of the holy grail. And our strategy was, okay, not to look at apomixis species, so other labs do that, but rather to take advantage of all the knowledge we were being accumulating about meiosis and reproduction in general to de novo engineer apomixis. So can we actually play with sexual reproduction to mimic apomixis? So we started with Arabidopsis, obviously, and to build apomixis, we need to target two things, meiosis and fertilization. Okay, so that's what I've just said. We need to target both meiosis and fertilization to get clonal reproduction through seeds. Okay, back to the schematic representation of meiosis, and we can summarize the differences between meiosis and mitosis in three features. The first one, which is very unique to meiosis, is to have two divisions following a single replication. The second is the presence of recombination, which is very high, shuffling chromosomes at meiosis. And the last one is the fact that sister chromatids, which normally at mitosis split from each other during mitosis, during anaphase, at meiosis one, they go together. This is unique to meiosis, to have the two sisters going together, and then the two homologous chromosomes are splitted rather than the two sisters. And this story started when we identified that mutant in the lab, which we called OSD1 for omission of second division. In this mutant, for the first time, we observed something striking, that meiosis did not produce 4 spores, but only 2. And we were able to show that in this mutant, what is happening is a single first division and no second division. They skip the second division and go directly to gametogenesis. So this is a representation of that. You have a first division, you produce diploid gametes, diploid spores, but the spores are recombined because we went through a first division of meiosis. So we targeted the first pillar, which is the presence of 2 divisions. Then we went for recombination. It was relatively easy. We know many genes which are required for recombination. We pick up the most obvious one, which is called SPO11, which is the enzyme that catalyzes double-strand break formation at meiosis. So without double-strand breaks, you don't have recombination. So we combine OSD1 with SPO11, and then we don't see any crossover anymore. Then you have 10 chromosomes which are not connected to each other because they don't have crossovers. But then you have missegregation of these homologous chromosomes and sterility. So we needed to combine with mutation that target the third pillar of meiosis, which is co-segregation of sister chromatids. And luckily enough, we knew one mutant which was able to do that. So we combined with the third mutation in the coenzyme protein called REC8. And when we combine those 3 mutations, we end up with this, which is we have alignment of the 10 chromosome, separation of sisters, production of 2 spores, which is mimicking mitosis. We call that mutant MIME because it's much easier to say than SPO11, REC8, or SD1. So now we'll call it MIME from now, which is mitosis instead of meiosis. Summarize this process for those who have not followed maybe. Combining those 3 mutations, we were able to turn meiosis into a mitotic-like division. And we confirmed that by genetics as well, that indeed we've turned meiosis into a mitotic division. Okay, wait a minute. We have abolished meiosis, transforming it into a mitosis, but we haven't touched fertilization so far. So what do we expect? We expect a doubling of ploidy at each generation. And this is exactly what we observed. Here you have a first generation MIME with 10 chromosomes going through mitosis instead of meiosis. The next generation, you have tetraploid plants with 23 chromosomes. They are still MIME, so mitosis instead of meiosis. Next generation, you have octoploid plants with 40 chromosomes. They are still MIME, but they are sterile, so I don't have the next generation to show you with 80 chromosomes. But I think I've made my point. Indeed, we double ploidy. So this is not what we want to achieve. These plants are genetically identical to the mother plant, but with twice the number of chromosomes. So we need to find a way to change that, to allows these gametes to make embryos without contribution of a male gamete. We were stuck. We had no idea how to do that at that moment. But then I met Simon Chan, who has just identified a line in Arabidopsis which was able to produce haploids when you cross it. So it's quite common. It's very known in maize, for example. It was the first line in Arabidopsis. It called that line gem. And the property of that line, when you cross it to another line, you have elimination of the chromosome of the gem line, and then you end up with haploid embryos. So the obvious idea was to combine MIME with gem. The prediction is that you will have clonal gametes, thanks to female gamete, thanks to MIME, elimination of the chromosome of the male, and then you should end up with clonal embryos, which have the exact same genetic content as the mother plant. We've done that. We have here a color code for the mother plant and the paternal genotype. And when we've done that course, we notify more than 30% of the seeds which were diploid with the exact same content, genetic content than the mother. So clones. For the first time, we are able to produce clones, clonal seeds in a sexual species. Okay, so summary, MIME plus male gem elimination, we get clonal seeds. Can we do that in crops? Because, of course, Arabidopsis is not really of interest economically, so let's go to a crop. Again, we pick up rice, because we have the right collaborator to do that, and we combine the same three mutations, abolishing recombination, cell cycle, modifying cell cycle, and changing chromosome distribution, and observed the exact same phenotype. The transfer worked beautifully. We have here the 12 chromosome surrogating and having a balanced surrogation at first division and producing perfectly balanced diploid spores. And we showed that those spores genetically are identical to the mother plant. So there's no surrogation, as you can see here, in a normal plant. Okay, so we were at the exact same point. We had these diploid gametes, and again we observed doubling of ploidy at each generation, and we needed a strategy to make those gametes going into embryogenesis and produce clonal seeds. And again, we met the right peoples. The first solution came from a paper published at that time in maize, where they identified the gene called maltrilineal, which is a gene behind the genome elimination line, which is well known in maize for decades. Then we thought that we can reproduce what we have done in Arabidopsis using that line in rice. And the team of Kejan Wang tackled that question, trying to combine MIME mutation with matrilineal mutation to trigger the formation of clonal seeds. And now we are in the CRISPR area. This kind of magic. We can make a quadruple guide versus those three genes. And in one generation, you can get quadruple homozygous mutants in hybrids that then produce clones. Bingo. They've shown that they obtain clones through seeds in rice. The only problem at the moment is that the frequency is relatively low. You have reduction of fertility due to the matrilineal mutation, and only a few % at the moment of the seeds are clones. But this is a proof of concept in rice. So summarize of that first strategy, matrilineal mutation plus MIME, you can get clonal embryos. And then second solution, we have MIME, we want to produce embryos. And a couple of years ago now, Sundar contacted me with it was just before Christmas, it was really a Christmas gift when he contacted me, saying that they have found something with Imtiaz in California here, or in Northern California, and they've identified BBM1 as a key gene for embryogenesis in rice. And the mechanism they have revealed is amazingly simple. Here we go. BBM1 is not expressed in the Excel. It's expressed only in the sperm cell. And the model was, when you have fertilization, the expression of BBM1 triggers embryogenesis. This is a very simple mechanism to prevent the egg cell to start embryogenesis before fertilization, because the key factor is provided by the sperm cell. The key experiment to prove that model was to express in the egg cell the BBM1 gene, which is normally not expressed in the egg cell. And when they've done that, they obtain a quite high frequency, up to more than 30% of haploid embryos through parthenogenesis. If you have followed me so far, the obvious experiment is to combine MAME mutations with expression of BBM1 in the egg cell. They've done that, again, triple CRISPR against MAME, plus transformation, plus introduction of the construct that triggers the expression of BBM1 in egg cells. And beautifully, they've shown that you can get up to 30% of the seeds being cloned. So you have no reduction of fertility in that line, and 30% of the seeds you obtain are clones of the mother plant. So I would say that with those two strategies, we are getting much closer than before to the holy grail of having apomixis in the field. So two strategies, MAME plus matrilineal, or MAME with BBM1. Today, MAME plus BBM1 is the most effective strategy, but let's see in the future if we can combine the two or improve both of them. Because frequency needs to be improved to a much higher level to be useful in the field. And very likely, because of conservation of the genes, very likely it will be easy to transfer that to any cereals, maize, rice. Rice is done already, maize, wheat, for example, or any cereal you prefer. And with this, I'd like to thank the people who have done the work. So most of that work has been done before I moved to Max-Planck now. So it was done in INRA, France, in Versailles, and the collaborators in Rice, in UC Davis, in Sierra in the south of France, and in China for the matrilineal story. And with this, I'd like to thank you for your attention.